provide my experience with the audience. Thank you very much. Thank you for the organizer on the drug discovery and design methods and applications. So today's I'm going to introduce you the world of docking. Here, what I'm going to do it majorly, I'm going to work out on. I'm going to give you a live example what we have done in our lab. So I have we have published. My team has published close to 30 papers, 35 papers, specifically only on docking, pharmacology, and relating with the GCMS data and compound analysis and toxicological studies. So, because we were recognized by DBT, our center, Department of Biotechnology and Bioinformatics Infrastructure Facility was recognized by Government of India. So we were having liberty to have a lot of softwares, the commercial softwares like Discovery Studio, among all. Then our lab facility has very good genomics facility. We have the sequencer with us. So today I'm going to discuss with the docking studies. What is the docking? What is the basics of it? So I got to know maximum students here, and we faculties are also students when we are learning. So please don't take me another way. So we all are learners. We all want to learn about what is happening in the world and how we can update our profiles. So let me start with our topic. I work on pomegranate. My major research is on pomegranate. I have published close to 20 papers, 18 to 20 papers only on pomegranate, specifically working on diabetes. So what is docking now? So I actually start with the basics always. So I'll start here. You can see here, a docking is nothing but a structure-based technique which attempts a best match between the two molecules. So you can see a puzzle we are solving here. And I'll give you one surprise number. There were 40,000 papers within Corona period published only on docking. Everyone was trying to suggest some or the other solution using In-Silico model. So you can understand the power of computer. You can understand the power of docking. Though it's little stringy to understand. So we should know the minimum criterias what we can use here in docking. So I am today going to introduce you people with those all criterias. And then I'm going to give a small case study what we have done in our lab, and it has got published in a journal called AICTE. It's called Experimentals and Clinical Research Related Journal. It's from Germany, so it's got published there. So now docking, you would have understood wherever we are matching two compounds, wherever we are there is the interaction between two compounds like protein and drug. You can say not a drug, it's kind of drug-like entity. When it matches with your proteins, it's interacting with molecule and give you some score and find a best match among that. That's called a docking. So what is really a docking? We all are comfortable with the enzymatic, the lock and key model, if you would have remembered that. So in the field of molecular modeling, this field called structural biology, this field is also called molecular modeling. If you go for dynamic study, it's called biomolecular and structure dynamics. So there are different kinds of words has been used for always molecular docking. Sometimes people do use the word called simulation also. So that simulation also can be used there. In the field of molecular docking, docking is a method which predicts the preferred orientation of one molecule to the other. So you can understand. So you can understand the perfect orientations. Perfect orientations means, if you remember lock and key model, where is the receptor and where is the molecule comes and bind here? Where is that getting a fixing? That's our orientation. So for that, it should move around or little it goes here. So those preferred orientation of one molecule to a second when bounds to each other and make a stable complex. That's called molecular docking. So now for this, we need to have a knowledge of preferred orientations. In turn, maybe it's used to predict the strength of association, binding affinities between these two molecules. Okay, one is called receptor, another is called ligand. So ligand is nothing but is a chemical entity that comes and bind with your receptor. Receptor, hope you know the receptor. Each and every cell has millions of receptors on that. So those all receptors, our cell, all the cells has the millions of receptors. So these receptors are different entities again. As example, you have epidermal growth factor receptor. You have tumor necrosis factor receptors. So your cell has lot of receptors on it, which get activated when a signal through brain comes upon. So this is ligand. Ligand comes and bind and start the process. As example, in the cases of EGFR, epidermal growth factor receptor, your tyrosine kinase pathway should start. Okay, it should get signal. So that's the way. Then RAS, RAF, MAP, kinase, all the signaling starts, and your mitosis, your process starts of that cell. Cell starts generating, that cell comes out, then localization should happen, then that localization if happens, then cells will get its nature, what cell goes to, where it should goes to. So this all is your mechanism. So whenever we are starting anything, we should be comfortable with the mechanism. We should know the positions. We should know the mechanism. So we should know what we doing. Okay. So minimum task is, so review of literature should be done properly here. So why docking important? Why you have to do the docking? So what is the reason everyone should do the docking? Look into this picture now. Just now I was speaking about the receptor. Okay. So look in this picture now. The association between the biological relevant molecules such as proteins, nucleic acid, carbohydrates, lipids, this plays a central role in signal transactions. Okay. Furthermore, relative orientations of two interactive partners. Look at this is here, the receptor with the adrenaline. Okay, adenine is again molecule, that is like a chemical entity. Okay, may affect the type of signal produced. It's called antagonism, agonism. So I think you all are comfortable with this old word, agonist, antagonism, antagonist. So there is, the docking is very useful to predict the strength and type of signal produced. So you can understand this. So this is the one pathway that you are seeing, the AMP pathway, cyclic AMP. He's here, as you can see here, this pathway. So you should know the protocol. You should know the mechanism behind it whenever we are starting studying. Okay, it could be used for any type of purpose. It could be used for environmental studies. It could be used for your disease related studies. So there are ways to look into your docking studies. Okay, so drug designing, why drug designing is very important. You know, the corona, now corona is on peak, so everyone wants to search a drug for it. So first they started with the computational biology. So computational biology saves a lot of time, a lot of period of searching a drug. So that's called rationally kind of drug searching. So we in turn to predict the affinity and activity of small molecules. So you can understand, so docking is frequently used to predict the binding orientation of small molecules. As example, you can see here is an orientation where we are looking for a small molecule drug candidate to their protein target. This is your protein target. Protein target uh, protein targets in order to in turn predict the affinity and activity of the small molecules. So because that you can understand now, docking plays a important role in rational designs of drugs. So now this is a small wording. Actually, this never been talked about. People usually go directly. So I just want to speak about a little about the glossary. I just wanted to speak about little glossary. What words has been used in the docking studies. First is the receptor, also called host, even also called lock. So this is a receiving molecule where your most receiving molecule where your ligand or drug comes and bind. So it is the most commonly used what most commonly used term where you can find a receptor structure. You can find a receptor structure in different kind of databases like protein data bank. Okay, you can always download a structure from there to work on the your receptor. So I'm going to show you a little practical after when my case study will I'll start. Then next it goes for ligand. Ligand is what? It's a complementary partner molecules which comes, binds and to the receptor. It when it binds to receptor, so ligands are most oftenly they all are small molecules, but it could be biopolymers also. It could be it could be it could also be another biopolymers. Okay. So next term comes docking. So what do you mean by docking? So docking is a computational simulation of a candidate ligand binding to a receptor. So docking. So you can understand a receptor and ligand binding is called docking. Next is called binding modes. So what do you mean by that? The orientation of the ligand relative to the receptor as well as the conformation of the ligand and receptor, which when it bonds to each other. So you can understand when receptor is a static form, when your ligand comes and bind here, it gives some or the other uh, answer. So that's answer we search in as a energy. We say as a energy. So energy, whatever energy volume comes out with that, we will predict the answer. So we will score it. We will try to score the molecule. Okay. So for that, where it goes and bind, that's called binding mode. Binding mode is you have different kinds of pose 1, pose 2, pose 3. So if you get a good score, does not mean your binding mode would be the first one or second one or third one. So here you manual check is needed. So when you do the manual check, that time you understand that your binding, your molecules had come and properly binded to the right place. Pause, I told you already, a candidate binding mode. Okay. Scoring, the process of evaluating a particular pose by counting the number of favorable intramolecular interaction. How scoring comes, how energy formulates. This will formulate with the bonding interaction with the hydrogen bonds and hydrophobic contacts. So this all comes under this way. Okay. So scoring comes out. So ranking, the process of classifying which ligands are most likely to interact favorably to a particular receptor. So free energy binding that comes with the ranking. So now, so type of docking problems, what major docking problems you will face. So one is come protein-protein docking. Next is come protein small molecule docking. You would have heard about SBD, structure-based drug designing, like LBD, ligand-based drug designing. So these are nothing but these all molecule only. So if I say protein small molecule docking, you can understand that a molecule comes and binding to it. This is called cavity, but it's behind scene. Here protein-protein docking means, so usually they use Hex software for it. So even commercial software is also available. So these two molecules go and bind with each other. Protein-protein binding will happen here. So protein small molecule binding, you can see here, you have a molecule and ligand comes and bind here. So you can understand with that. So definition, the problem definition. What is the definition of a problem? So you can understand, one can think of molecular docking problem as a lock and key molecule. What is the lock and key molecule? So you want to find a correct relative orientation of key which opens up the lock. So please understand one thing, if you have a wrong key, your lock will not open. Hope that would be clear with everyone. So you need to search a proper molecule, a proper lock, a proper key to connect with each other. So the directions also need to be searched for. So you can understand as an induced fit model. Hope you all know about the induced fit model. You would have heard about induced fit model. So what do you mean by induced fit model? Induced fit model is the both the ligands and proteins are flexible. You can understand your hand, if you put one glove in your hand, okay, and the analogy will come as a lock and key. So during the course of docking process, the ligand and protein adjust their conformation to achieve the best fit. If I say two molecules are coming together, it should fit best. So that analogy should be seen. So best fit should be seen here. So conformation or adjustment result, it will result the overall binding is referred as an induced fit. You can see in the picture, this molecule comes and bind here. So if it will fix it here properly, you can say the substance entering the active site of the enzyme, and that clamp is around the substrate, forming an induced fit. So this is one example of your definition you can see here. Next, so what is docking approaches now here? So first is called a shape complementary. So one approach where it's a machine technique again, where it describes the protein and the ligand as complementary surfaces. Simulation studies, already I told you, the interaction, it is complementary scores, whatever comes out. The second approach, simulate the actual docking process in which the ligand protein pairwise interaction energies are calculated. So all the energies are calculated. Both approaches have significant advantage as well as the limitations. You can understand what the limit. How this score comes, already I have spoken about it, but I'll tell you now. The bonding pattern. The bonding pattern is you have hydrophilic, hydrophobic, hydrogen bond donated, hydrogen bond accepted, hydrogen bond. So this is there is one rule we do follow, that's called R of 5. It's called Lipinski rule of 5. Rule of 5. Lipinski rule of 5 is used here. So there are specific rule given to approach this to understand the ligand. So binding affinities, ionic interactions, aromatic interactions, van der Waals forces, electrostatic forces, free energies. So look into this example now. So this is your HIV protease target. One example I have seen here. So shown here. So aspartate group is the active site. So how we got to know this aspartate group is the active site? So for this, you have to go through some literature and you have to see which molecules are taking a proper uh, part to infect the organism or to the host. So you have to look into that. Then after you go through with the research papers and all, we got to know aspartyl groups are in active site. So you can understand. So now you have to generate a molecule surfaces for the receptors. After generating, now the active site. Look at the active site. So with the active sites of the receptor, look into that. The spheres become potential location for the ligand atoms. See now the spheres. So what do you mean by the active sites? Active site is nothing but a cavity found on the receptor where your ligand goes and bind. So active site is can be predicted. There are a lot of databases are there. There are a lot of softwares are there. So you can say binding DB. So binding DB is one. Binding DB is there. Binding DB can be used. There is cavity prediction tools are there. So there are a lot of tools are there. These all tools I'm going to show you next when I'll follow up with my slide. So then next what we do, step 4 says we look into that ligand atoms matching. So determine the possible orientations of the ligand. We will look into that orientations. After you see the orientation, then you find the top scoring match. Whatever top scorings are there, the top scorings scheme. With the top scoring schemes, the shape we will see electrostatic scoring, force field scoring. So this all should be the score. Look into that for the best matching. So next, so what is the applications, the major application of molecular docking? What would be the major applications of molecular docking? You can first we can determine the lowest energy structures. So I told you the best scoring. Best scoring we will find it. We will always say if it is having a lowest energy consumptions. So lowest free energy structures for the receptor ligand complex. Then we calculate the differential binding of a ligand to two different molecules, macromolecules receptors. So we can calculate those all. Okay. Then even you can study the geometry of a particle complex. Okay. So you can You can modify your ligand again and again, and you can optimize the potency of that. As example, you can, very simple example you can think of. I'll give you example of benzene ring. If benzene ring, if I'll put one OH, it acts like a phenol. If I put NH2, it becomes amine. So this one molecule, if I will use it to dock it and check the scoring pattern, then I will change this R group, as said, NH2 or CS3. I will do CS3 or CHOCH3, means ester we will make it. So, or ether we will make it. So as example, the group changing, or we will put it as a COOH there to our benzene ring, R group formation we are changing. So that geometry modifications, we can always use it to optimize the potency and other properties. So next, de novo design for the lead generations, we can do it. We can generate a library. That library generation, you can do it. You can create a database over it. So we have created a database called phytomeliters. In that we have put it all the molecules whatever has been best stocked for diabetes. Okay, even now we are working for another database for the on the computational biology, using computational biology. So you can design a library for your own molecules. You can screen the side effects because which will be caused by the interactions with other molecules. That can be done with a toxicological analysis. ADME can be can use for that. So there are different kinds of tools are there. Okay, so even interact with the interaction also, how, what is the scoring will be there. With the basis of that, we can check the toxicology to check the specificity of the potential drug against the homologous proteins. So you can check it with that. You can even docking now is a widely used tool for the protein-protein interactions. There are several things people are looking for. Okay, then okay, so then uh, knowledge of the molecular associations with variety of pathway you can work out on that. Then docking can be used to predict the pollutants that can be degraded by the enzymes. So now the component, this is common softwares. The common software used for the docking purpose, you can see here. First is the doc USA. This is the software now freely available. Even for this you should have your university license, your mail ID. So for that you can always use it. UCSF doc software is a widely used software. Then another widely used software is freely accessible. It's called AutoDock. If you know how to run AutoDock, you can do anytime docking studies very easily. So it's a freely available tools, widely used tool. Then there another variant of AutoDock is available. AutoDock 4 and AutoDock Vina is available. There are two generations of these softwares are there. AutoDock 4 and AutoDock Vina. You can look over in that AutoDock, and there is manual available. These all softwares has their help manual available in their online. So you can always use it. Okay. If then also you're facing problem, we widely use AutoDock, and then we use Discovery Studio. So if you face some problem, you can always come back to us. FlexX, another software. It is a highly used software. Okay, so what is the AutoDock? AutoDock is a basic software to use small molecules such as substrate or drug molecules that bind to the 3D structures. Okay, this is the website of AutoDock, autodockscripts.edu. The next is the Flex, is from Biosol YT. Okay, here the rigid docking can be done. Receptor is treated as a rigid, and then they use the algorithm called incremental constructions algorithm. So break ligand up into the right fragments and then try docking it. Then whatever result come, and then they confirm with the energy confirmations will be done. Next software is used as a GOLD. Gold is a genetic algorithm software. So its name is that's why it has given name as a gold. Gold is called genetic optimizations for ligand docking. So you can go to their website. You can always look for that. Okay, you can always look for that. Okay, see performs, then it is automated docking software. Okay, automated docking with a full acyclic ligand and flexibilities. Okay, So like in flexibilities can be seen there. The score in this, it includes the scoring. Uh, it includes H bonds scoring on the basis of hydrogen bonding and the hydrophobic interactions, mechanics and molecular internal energy too can be seen here. So there are basics. Other softwares are also available. One is called AADS. It's from India. There is Dr. Jayaraj is working in IIT. Professor Jairam. He is well-known computational biologist in India, and he was the person who came with a proposal called open source drug discovery. Okay, and with Professor Raghava, who's in this their Bhatnagar awardee. Okay, so is the one of the highest award given to the science in the science our life sciences field and sciences pharmacy or other fields. Okay, but Nagar awardee there, and he is working in IIT Delhi, and Bhargava, Dr. Ragwa is in working in triple IT, okay, Indraprastha University. So these both are the highly influential people in computational biology and known for their software development. And Dr. Agwa comes in top 10 cited people in the world and top 10 highest uh, database creator in the world. So he's the one from India. So you can always look for his profiles and all. You will learn, you will understand there are a lot of softwares they really keeps on making, databases they keeps on making. Okay. Then another software is called ADM from Japan. Then Vitadoc, South Korea, Darwin, USA. Then even you will find another docking analysis software is Diwali. Okay. So then doc version from Canada, Glide from Schrodinger, and BioWire Discovery Studio is from X-Release, and now it's called uh, Dashoot. Okay, so this software BioWire, but Discovery Studio I work on a lot. So this is the major softwares, but these are the commercial softwares. So free softwares already I told you the Indian softwares, Sanjeevani's and all the things you can find it. Even AutoDock is free, and there are other softwares also. The DOCK doc software is also there. These all softwares needs, but it needs your academic IDs. Okay, so now I come to the major what we are going to learn about the case studies. Okay, before I go to next, I just want